are in the third video of the chapter 11, that is Biotechnology Principles and Processes. In the first video, we had an introduction to the entire process of biotechnology where you looked at all the steps. Second video, we looked at the how to extract DNA from an organism in which you have identified your gene of interest. Third video is this one wherein you should understand how to isolate this gene of interest from the extracted DNA. If you're unable to follow, please watch the previous two videos so that you get the flow of events. Okay. Now, I have got my DNA extracted in the purified form. I know that my gene of interest is present in that DNA. I need to cut out only that part. I don't want the entire DNA. All right. I just need that part. How to get it out? I can't pick it out, obviously. So I need to use some scissors called as molecular scissors. Now, as you know, molecular scissors, when we talk about, you should not think of actual physical scissors. They are enzymes. Okay. So enzymes. What kind of enzymes are they? They are called as, let me type it better. Yeah. They are called as restriction enzymes. Okay. Restriction enzymes. What are these restriction enzymes? They are nucleases, meaning nuclease will cut a nucleic acid, protease will cut proteins, and so on, right? So these are nucleases. Nuclease can cut a nucleic acid. So it will definitely cut a DNA. Where does it cut? That will depend upon what kind of enzyme we have picked up. So there are two different types of restriction enzymes. The first type is called as the endonucleases. Endonucleases, one word. I'm just splitting into two so that you understand. Endo usually means what? What is the word? Endo means from inside. Right? The same way, exo meaning from outside. What are they doing from inside and outside? They're going to cut our DNA. How are they going to cut? Now let us say this is my sequence of DNA. This is my DNA. All right. Double-stranded DNA. Nice one. How is this one going to cut and how is this one going to cut? Endonucleus, since it is inside, endo, it will cut from within like this, here and here. What is it cutting? It will cut your phosphate sugar bond. I hope you remember in the DNA ladder, there is the pink line that you can see is made up of sugar and phosphate, and whatever is protruding inside to this part would be the nitrogenous bases. I hope you remember that much. Yes. All right. So this endonucleus will cut within from within. It will create such small fragments after it cuts. This is the cut, right? It will cut off like this. I'll explain it in detail, don't worry. So this is going to cut from within the DNA strand. On the other hand, you have exonucleases. So where is it going to cut? It will cleave at the edge or the tip. It cuts here. One nucleotide it is removing, one nucleotide it's removing, like that. Hope it is clear. So you have two different types of two different types of restriction enzymes called as endonucleases and exonucleases. When was this found out? When did they find out such a thing existed? They discovered this in 1963. Hope you remember. I want you to remember this because usually they'll ask you in either MCQ or they'll ask you to give a short note on restriction enzymes. You should be able to write. Okay. In which organism did they find it out? In E. coli. Please underline when you're writing by yourself. Since I'm typing, I've typed it like this. Escherichia coli. So two things. 1963, Escherichia coli, they found out there are enzymes which has the ability to cut DNA. How many types did they find? They found out two types are there, endo and exonucleases. How does endonuclease work? Is your next question. Exonuclease, what does it do? It just cuts from the corner and from the ends of the DNA. Endonuclease, you will get question. You will get question and you should be able to answer. How does it cut? First step. So you will write it in this form of steps. All right. So how does endonuclease work? When you get a question on this, you should be able to write first point. It inspects the DNA. What is it inspecting for? To find specific sequences. Inspects the DNA for specific sequences. Sequence of what? Sequence of nitrogenous bases. Let us say one sequence is there on this DNA. Okay. So I'll just draw this here in a larger form so that you get what I'm talking about. Here, let us say G, A, A, T, T, C is a sequence. Let us say, just giving one example. So this is a sequence for one particular restriction enzyme. Its name is EcoR1. Okay, some name. I'm going to teach you how do we name them as well. All right. So what does it do? It first and goes and sees for on the DNA, where is the sequence? Where is my specific sequence? It is going to be called as recognition sequence. What are we going to call this specific sequence? We are going to call them as recognition sequences. Recognition sequence. Recognition sequence of one restriction enzyme will be different from the other restriction enzyme. Please remember that. Once it finds out, okay, my recognition sequence is here. What does it do? It goes and binds over here. After binding, it will cut the DNA. Where does it cut? It cuts on the sugar phosphate bond. Where is sugar phosphate? Where is sugar phosphate? Now, I have written GAATTC on the line. Is it correct actually? It is not correct, right? I should not write it over here. Where should I write instead? If I'm drawing the DNA like so, the sequence will be G, A, A, T, T, C. It's protruding, isn't it? It should protrude. Inside the ladder it is. Inside the ladder, like the steps of the ladder. The line that I've drawn in green is what part? The sugar and the phosphate. I hope you remember the structure of DNA from our chapter 1 or chapter 2. Please go through it one more time if you don't remember this. Okay. So it cuts the sugar phosphate group. Sugar phosphate bond. Things to remember again, revising. Restriction enzyme. Why are we learning about restriction enzyme? We are learning about restriction enzyme because it is a tool in genetic engineering. Where is this tool used? This tool is used to isolate our gene of interest. We need to get our gene of interest out from the extracted DNA. For that, the tool that we usually use is an enzyme called as restriction enzyme. There are two different types of them, endo and exonucleases, or restriction endonucleases and restriction exonucleases. Clear? Students, let us understand the action of restriction enzyme, specifically restriction endonucleases. So what does it do first? We already discussed what does it do. It looks for its own recognition sequence, which is a specific sequence, which is not the same for two different restriction enzymes, right? Now, where is it looking for? On the DNA strand, it is looking for both strands will have that sequence. And there is one speciality about that sequence. Okay. So let us say in this case, the example given of the restriction endonuclease here is EcoR1. The name of the restriction endonuclease is EcoR1. It looks for sequence called as GAATTC. Where is it? Now this is your double-stranded DNA, isn't it? Double-stranded DNA it is. Now it is going to be continuing like this, but we are not taking that part. Instead, what are we doing? We're just opening it out. We're just opening our DNA out. All right. This part, instead of the curve, we're just opening it so that we understand what is happening. All right. Okay. So here, protruding inside is your GAATTC. What is it? It's the sequence of the nitrogenous bases. As you can see, this GAATTC is found twice, meaning it is starting from here GAATTC on the sequence. On the other strand, this sequence is repeating in the other direction. Okay. Such sequences we also call as palindromes. Palindromic sequences. Palindrome. You know what is a palindromic word, right? if you write M-O-M, mom, you can read it from this end also, you can read it from this end also. Same way, dad, mom, such words are called as palindromes, right? So here, these recognition sequences of restriction endonucleases are palindromes. Remember that, please. Okay. So where does it cut? It doesn't go and directly cut starting from its sequence. It doesn't cut here. Instead, where does it cut? It cuts slightly away from the center. Where is it cutting? It's cutting here. It's cutting over here. This after G, it is cutting here. The same way, here there is G, A, A, T, T, C written. It cuts over here. Clear? Right. So when it cuts like that, when it cuts in this pattern, when it is cutting, what does it create? It creates something called as sticky ends. I'll just show you. I'm erasing the other line so that you can see where is it cutting. It's cutting away from the center. This is the sequence GAATTC, GAACTC. On both strands it is present. This is a sequence. It is not going and cutting at the center. It is not going and cutting at the starting of the sequence. It's cutting little away from the center, which is here is the center. So where is it cutting? It's cutting here, and it is cutting on the other strand right after G. Now when it cuts like that, what will happen? It will create sticky end. It's called as a sticky end. You can see in this picture, next picture. This is the cut that is made. Now you have some protrusion. This protrusion is called as a sticky end. This is going to be of high significance for us when we are going to put this gene inside a vector about which we will be learning. All right. So things to remember when it comes to restriction enzyme. The year of discovery, 1963. They are nucleases. What is nuclease? The one that can cleave nucleic acids, right? They are of two types, endonucleases and exonucleases. It was first discovered in E. coli. It has specific sequences called as recognition sequences. What is a sequence of action? First, it inspects on the double-stranded DNA, looks for specific sequences called as recognition sequences. Next step is binding. I didn't write it. It's called as binding. It binds on to the double-stranded DNA. Once it binds, what does it do? It cuts. How does it cut? This cuts, it looks for its palindromic sequence or the recognition sequence. After that, it cuts slightly away from the center of the sequence, resulting in sticky ends. This much when you write, you get 2 marks. That is clear. Now, we are not done with restriction enzymes. Let us continue a little bit more of data that you have to remember when it comes to restriction enzymes. Okay, so what is the other thing? The first restriction enzyme that was discovered was called as one was called as Hind, Hind, Hind. Now, why this weird name? How to name it? All that we will learn right away. It is called as Hind2. The first restriction enzyme that was discovered was Hind2. What does it recognize? How many base pair does it recognize? You should know. It recognizes a 6 base pair sequence, just like our GAATTC, 6 base pair sequence. Just like that, this also recognizes a 6 base pair sequence on the DNA, but it is not GAATTC. It is different, but we are not learning that because it's slightly bit complicated. Okay. How many restriction enzymes till now has been discovered? 900. From how many organisms? 230 strains of bacteria. This much data you need to remember for your exam. Right. Now we'll learn how to give a name to the restriction enzyme. So the example that you saw, how does an nuclease cut a DNA? You saw this name EcoR1 and I told you that I will explain what does that mean, right? So how to name? How did this get this name EcoR1 from where, right? So from the organism from which it is discovered, the name of the organism was Escherichia. You have to learn this, children, because with this example, you are going to explain naming. Escherichia coli, which is the Scientific name. Okay. Scientific name usually has a genus part, which is this, and the species part. This is the genus part, and the species part is here. We should also know the strain of the organism. Strain meaning what? Strain means a subtype of the species. For example, in our coronavirus itself, we had been talking about delta and alpha and the killer one and all those things, right? So those we called as the deadly strain and harmless strain. I hope you remember. So strain is just a subtype of a species. So here in this organism, the name of the strain was RY13. Right? So now look at the naming of this EcoR1. Where did this E come from? From the genus, first letter. From the species, two letters. From the strain, one letter. Yeah. So we got E, co, R. Now there is a Roman letter after this. Where did this Roman letter come from? This Roman letter is the order in which it is extracted. So from this organism, first order, this was the enzyme that was extracted. So we call that, we number it as 1. I hope it is clear. We'll continue with gel electrophoresis in the next video.